Good afternoon, everybody, and welcome to my live-streamed presentation entitled Hunt for COVID-19 Vaccine. My name is Bernd Rehm. I'm Professor of Biotechnology at Griffith University, but I'm also the Director of the Center for Cell Factories and Biopolymers, embedded within the Griffith Institute for Drug Discovery at Griffith University. Before I continue with my presentation, I'd like to thank all past and current members of the Center for Cell Factories and Biopolymers. I'm privileged to work with talented people who are dedicating a lot of hard work to the progress and science we are making and to help developing technologies which provide solution for global challenges. We are an interdisciplinary group. We have members with qualifications in chemical engineering, biochemistry, microbiology, immunology, and our interdisciplinary nature of research and development activities require us to collaborate with national and international institutions. So we are highly networked, working with institutions across the globe to develop disease-relevant solutions, vaccines in particular, but also to tap into technologies to characterize our materials as best as we can. I also acknowledge funding bodies on the right bottom corner. I'd like to emphasize two people in my team, and this is Dr. Shushung Shen. He is the lead scientist working on our COVID-19 vaccine at the moment. He is driving the research and development activities, and he's supported by PhD student Ben Everett, who's helping with the animal trials. So if I'm going to the next slide, the content of my presentation is depicted here. First, I would like to talk a little bit about our overarching research strategies and objectives within the Center for Cell Factories and Biopolymers. And then I will quickly move into a rapid response vaccine platform technology we have developed over the last few years. And we always had a strong interest in technologies which can be quickly adapted to emerging pathogens. So they need to be flexible, they need to have a platform character, and they need to be fast and leading to manufacturing at a cost-effective level. And then I will talk about the COVID-19 vaccine development activities. There's a lot happening in the COVID-19 space, as you can imagine. It's of such global impact that many research institutions and private industry are heavily engaged to find solutions. So the next slide shows you quickly our mission. The center is aiming at providing solutions for global health and environmental challenges by creating innovative functional materials and by developing platform technologies. We try to achieve that by harnessing the capacity of biological systems, so tapping into nature's creativity and capabilities to synthesize and assemble biologically active materials. We are applying synthetic biology and biotechnological approaches to then actually design materials to order and to also develop manufacturing processes which are required to be aligned for future applications. So the next slide gives you a schematic outline of our research strategies and what we are trying to aim for. The research strategy is depicted here on this slide, showing you that we are applying synthetic biology and bioengineering approaches to utilize the biological cell and predominantly the microbial cell as a factory. We are reprogramming microbial cells to connect biological building blocks in order to achieve functional materials. And those building blocks are predominantly proteins and biopolymers. And as we all know, these building blocks can be enormously diverse, and hence the design space to make advanced functional materials is tremendous. These materials can show performance which make them suitable for applications, and that's shown here. We have been looking at a number of applications, looking into bioseparations, protein production, biocatalysis, diagnostics, etc., drug delivery, and of course vaccines. We are talking to end users, to industry, to other researchers, disease experts, to understand unmet needs. So we are driven by unmet needs and specifications required to solve those unmet needs in terms of tailoring our approach, making materials that can provide solutions. If we identify prototypes, and not only those that show proof of principle performance, but those that meet all the criteria and the robustness of like a commercial product, we take those prototypes and then apply our development strategies. That means we are taking the prototype through manufacturing process development, product characterization that includes quality assay development, that includes characterization of the product, that inherently leads to intellectual property, secret know-how, and a potential commercial outcome. We are focusing on vaccines. That's a major effort of the research group of the center, and we are interested in platform technologies as previously mentioned. I will focus in my talk on one of the most advanced platform technologies we have developed in my lab and in the center, and this is related to vaccine particles assembled inside microbial cells, and in particular bacterial cells. We are making discoveries, we are discovering new materials, we are matching the performance of new materials with possible applications to harness the competitive advantage. Not every material will solve every problem. You have to be smart about how to actually further develop a material based on its properties. So we are translating those discoveries to solve unmet needs. And these are just a few examples of nanostructures, nanoparticles you can find in nature. And we have an interest in nanoparticles and microparticles. They are, by their size, mimicking a potential pathogen, and hence they are potentially good candidates for vaccine delivery strategies. There is one particular assembly you can find in microbial cells, and that's shown here at the right bottom corner. And these are naturally occurring protein-coated polyester beads. They are formed by a large variety of bacteria. It's a natural storage granule. And I will take you through the discovery and the actual platform technology development in a minute. But why are we focusing on vaccines? Vaccines are considered as one of the major breakthroughs in medicine, preventing millions and millions of deaths by preventing infection by pathogens, by viruses and bacteria. So here's a number showing you from 2010 to 2015, it has been estimated that vaccines prevented more than 10 million deaths. So you can see the impact of the relevance of vaccines in preventing infectious diseases. Smallpox has been eradicated and polio has been eliminated in many countries. There's still an unmet need in the vaccine development space. As you can see, infectious diseases are still contributing to 26% of all deaths in the human population. So why is there an unmet need? There are still no vaccines existing for numerous pathogens, viruses like hepatitis C. Of course, we know the coronaviruses, SARS, MERS, many bacterial pathogens, emerging pathogens, mutating pathogens. I should mention here that increasing antibiotic resistance makes prevention even more relevant because usually or before harmless infections become untreatable, hospital-acquired infections become untreatable, and hence prevention is becoming more relevant. There are also existing vaccines with poor performance. They are not very efficient. The rate of protection is pretty low. They are too expensive, so they cannot be used in wide vaccination schemes. And they show poor stability, so they cannot be really stockpiled to respond to emergence of pathogens. So there are properties which also justify vaccine development. And of course, I should mention animal health. In animal health, we have a need for vaccines to support food supply. Animal diseases in agriculture are making a significant economic impact, and there is a need and a requirement to develop further vaccines. Of course, cost of goods are major drivers in the animal health space, but also we should here notice that a number of zoonotic diseases are spreading from animals over to humans, and that's considered within the One Health aspect. So we are keeping animals close to us healthy, avoids us to become infected by certain pathogens. So if we are looking at vaccines and what has been historically licensed by regulators in, for instance, the US, the FDA, we are talking about the whole cell, whole virus vaccines. They can be live, but then they have to be weakened and attenuated. And a well-known example is a vaccine against tuberculosis, the BCG, which is related to Mycobacterium tuberculosis, the actual pathogen, but not identical. So it can be used as a vaccine. It has been traditionally used and it's still used. Inactivated vaccines, a very famous example is the polio virus, has been invented many years ago and is still being in use where relevant. Subunit vaccines have been recently emerging. They are basically depicting individual components of the pathogen. They are safer than the whole cell, whole virus vaccines, but they are less immunogenic, and one has to be very careful to select the right antigens. But they are very defined vaccine formulations, and hence they offer a great opportunity for very specific immune response. They're very safe, and hence provide an opportunity for vaccine development. And then also the toxoid vaccines, they have been licensed. They are triggering an immune response which neutralizes the toxins produced during infection. So we have heard in the news a lot about DNA and RNA vaccines. They have been very recently emerging as novel vaccine platforms. DNA and RNA encoding antigens of the pathogen of the virus are being used for vaccination. They cause transient production of the antigen encoded by themselves, and hence induce a very specific immune response. Though those have been now in clinical trials, but there are no licensed products on the market yet. Another space for vaccine development that's depicting the area where we are focusing our efforts, and that's around formulating subunits or defined antigen formulations into particles, self-assembling them into vaccine particles, and using the mimicry by size of a vaccine particle to induce a strong immune response in patients. So the vaccine particle mimics the size of the pathogen that triggers a series of immune response mechanisms. I will talk about those in a minute as well. So now I will talk about the vaccine the platform technology we have been developed over the last few years. We have a focus on that technology. It's most advanced, and it is based on the ability of microbial cells to produce polymer inclusion. So this is an electron microscopy of a bacterial cell which was well fed with sugar, So it's actually producing these intracellular inclusions, these polymer particles. And these polymer particles fill up the cell, and then the cell is filled up, and it's, it's full of energy and carbon source because the polymer is a storage material, it's a reserve material. At the time when I was looking at these cells, everybody thought that if you isolate these particles, they would quickly collapse and would lose their structure. So it was pretty much by serendipity that we discovered that they can be propagated outside the cell as stable structures, because we looked at very old samples at a time, a few years back, old samples we wanted to clear out of the fridge. And I told my student, hold on, don't throw them away. Let's have a look through the microscope. And that's what we did. And we found these inclusions were beautifully shaped and spherical and didn't lose their structure at all, even after many years being stored in the fridge. And that triggered a whole lot of ideas and concepts on how to actually bioengineer these particles to incorporate proteins into its shell. You can see the more electron-dense boundary of those inclusions, and that's the protein layer. And we then came quickly up with the concept of incorporating antigens, so pathogen-derived antigens, into the surface of these polymer particles, and that was all being integrated into the microbial cell, then being able to produce those polymer inclusions, but coated with antigens of interest. So if we now look at the next slide, and that shows you the actual strategy on how we develop a vaccine based on those polymer particle inclusions. So what we do is, and there's a lot of effort required and smartness required to identify suitable antigens, because remember, these are subunit derived antigen formulations. So we have to look at specific antigens which are relevant to the pathogen and also relevant for the induction of protective immunity. So we look into bioinformatics. There are algorithms available to predict epitopes, but also we are looking into immune responses in patients. We're looking into immune responses in patients that recovered from the disease. So we know there have been established immune responses that mitigate the infection and allow clearance of the pathogen. That allows us to do reverse vaccinology and identify antibodies directed against certain antigens, and those antigens we are using to incorporate into the cell factory. So we do bioengineering to enable attachment of selected antigens, and it can be multiple antigens at the same time, to be attached to the polymer inclusions. Once we have engineered the cell factory, it can be endlessly propagated and grown up to high cell density fermentation. So we generate a lot of cell mass and then use a biomanufacturing process to retrieve the polymer particles coated with the antigens of interest. And here on the right-hand side, you can see the product after isolation from the cells. These are the spherical particles. They are coated with the antigen, so they are mimicking the pathogen by size. And they're also mimicking the pathogen by showing the antigens at their surface. But they are completely disconnected from the pathogen. They are not self-replicating, and they are extremely safe. If we look into the polymer itself, the polymer has been approved by the FDA for clinical trials. It was used in medical implants, and the polymer degrades inside the body after vaccination into 3-hydroxybutyric acid, which is shown here. That's part of the polymer. It's a constituent of the polymer. It's a homopolymer. That's the only constituent which is connected to form a large chain molecule. And this monomer is also found in human blood. So the human body is very familiar with the degradation product. So we have extensively studied different prototypes of these particles with different antigens and extensively tested their performance in a range of different species, large animals, cattle, sheep, deer, mouse, rat, guinea pig, in the context of different infectious diseases, in the context of bacterial pathogens and viral pathogens. And we found that they were safe as they were administered and induced significant humoral, meaning antibody responses, and at the same time cell-mediated immune responses, which is in particular relevant. The cell-mediated immune responses are in particular relevant for viral infections. So we correlated the immune response with protective immunity by doing animal challenge studies, etc. So I have to move on because I think I'm slowly running out of time. So this shows you the timeline of development. As soon as we identify a pathogen sequence, we can convert that information into producing a vaccine candidate through our cell factory principle. We do animal trials to test the performance of the vaccine candidate, and then we do manufacturing at a high standard to obtain a clinical vaccine material, which can then be tested in humans. So the pipeline is indicated here, it's less than 4 months. So this platform uniquely combines a very vast design space, meaning we can adopt numerous antigens and display them together at the same time on the same particle, with outstanding vaccine performance. That means induction of immune responses relevant to protective immunity, and we can cost effectively manufacture at scale. So the platform has been tested. The true platform character has been tested by developing, as mentioned before, different prototypes in the context of different diseases, and they're all shown here. And in all these cases, we have induced protective immunity in animal models, showing that the technology is a platform technology which can be adapted to different diseases. What we could demonstrate is that it offers a unique design space for the development of multivalent vaccines. So not only one subunit antigen component can be incorporated, it can be multiple, which makes the response more specific and also stronger towards protective immunity. We induced both antibodies and T cells, which is again very important in the context of viral diseases. And we have demonstrated that by producing numerous prototypes and extensive clinical data. So how do these polymer particles actually induce immunity? We are still investigating that in more detail, but the current unpublished data give us a very good idea on how they work, and that's shown in this slide. If we take polymer particles which is decorated with antigens of interest, in that case virus antigen-coated polymer particles, they are actually quickly taken up by the sentinels of the immune system, so the gatekeepers of the immune system, the antigen-presenting cells. They are taking up those particles very efficiently, and I show you in a minute images, and then they digest these particles and present parts of the virus components, parts of the virus antigens on their receptor molecules to communicate with T helper cells. They pick up the information, they get stimulated and specifically expanded to induce in this direction and induce and stimulate B cells that are then producing antibodies. And those antibodies can then neutralize the virus by blocking entry into human cells, and at the same time also tag the virus to enhance its uptake by immune cells and its destruction. This pathway shows you the cytotoxic T cell response. This is a response which enables the cytotoxic T cells to look out for infected cells and destroy those infected cells to further reduce the viral burden. On the left-hand side, you will now see an electron microscopy image showing you those antigen-presenting cells. And they are migrating through the lymph nodes, and they are present around our body to quickly take up a pathogen and pass on the information to the immune system. So if we are adding our vaccine particles to those dendritic cells, these are human dendritic cells, they are antigen-presenting cells, they quickly take up our particles, and that's the first step in mediating a strong immune response. And the more efficient the uptake is, the more you get a prolonged display of those viral peptides and a prolonged and extensive induction of an immune response. So the competitive advantage of the polymer particle-based disruptive vaccine technology is summarized here. I'm not going through that in more detail. I might just mention in addition that we see enormous shelf life. The particles can be formulated into powders, and then only prior to use they need to be rehydrated, and that tremendously adds to stability at ambient temperatures. We can quickly develop and can produce at scale cost effectively. We are using the platform to precision engineer a COVID-19 vaccine, and the precision engineering is aiming at inducing immune responses which are now emerging from the literature. So we are looking at tailoring a vaccine to induce responses we can find in patients that have quickly recovered from the disease, so reverse vaccinology, but also we are using historical data around the SARS vaccine development activities. SARS, the related virus to SARS coronavirus type 2, caused an outbreak in 2002 and 3, and there have been follow-up vaccine development activities. So now let's talk about the COVID-19, and I looked up this morning the tracker from the Johns Hopkins University, and it said total confirmed cases are now beyond 4 million, and the total deaths are reaching 285,690. This is of course a massive global impact, and as we all know from news releases, continuous news releases, that this disease is impacting humans everywhere in the world, has tremendous economic impact, and is an unprecedented pandemic threat. It's caused by this virus, which is very related to the SARS virus, as I was mentioning before. It's established now that this virus binds to a receptor, the ACE2, in cells of the lung, heart, and other tissues. It's causing pneumonia. It's causing, under some circumstances, linked to a severe outcome of the disease, a cytokine storm. It's a dysregulated immune response. Basically, it's an overreaction of the immune response, causing inflammation and destruction of the lung tissue. And that's one of the major reasons of mortality. And as mentioned, more than 4 million cases. Looking at the mortality, it's ranging quite substantially between the countries. Australia is at 1.4% mortality, is doing very well. But other countries like Belgium, they have a mortality of 16.4%. SARS itself had a mortality of, the outbreak in 2002, 2003 only had a mortality of 9.5%, and it was rather contained, only infected roughly 8,000 people. So that's the virus. The virus is an envelope virus. Its genome is an RNA-based genome, which is part of the core, and the genome is attached to a nucleocapsid protein, which protects the genome and makes up the core together with the RNA inside the virus. It's an envelope virus, so there's a membrane surrounding the virus, and embedded in that membrane we have numerous proteins. We have a membrane glycoprotein, we have envelope proteins, and we have the spike protein. And the spike protein seems to be a very critical target for vaccine development. So I will come back to the spike protein in a moment. The replication cycle is pretty well understood. The virus attaches to a cell and then it's endocytosed, it forms an endosome. The spike protein is very critical. It causes a membrane fusion. Genomic RNA is released. We have some protein production. We have recruitment of the host cell machinery to fire up replication of the RNA genome, the viral genome, and then genomic RNA is produced. At the same time, we also get transcription and translation. So meaning that viral components, the spike protein, the membrane protein, and the envelope protein are produced, and there's a self-assembly process, membrane engulfment, leading then to formation of the virus and release of the virus. The spike protein is shown here. That's a very recent publication, as you can see here, published in March in Science. It's a beautiful structure, cryo-electron microscopy. It's a trimeric structure. I'm not going into detail. It's very similar to the SARS spike protein. And as you can see here in green, that's the receptor binding domain. That's a very critical domain which interacts with the receptor, the ACE2 receptor, so mediating the binding to the cell surface. This is now here in the up position and can interact with the receptor. If you look at the right upper corner, you can see now the red dots in that receptor domain. That's the structure of that receptor domain, receptor binding domain. And the red dots show you where the new coronavirus, the 2019, so the COVID-19 causing virus, differs from the previously occurring SARS virus. So you see within that receptor binding domain, we have a number of red amino acids which are contributing to the binding to the ACE2 receptor. And they actually cause a substantial increase in affinity in binding to the receptor, and that has been also attributed to its success to actually become a global pandemic. Only a few viral particles are required because of that high affinity to attach to the surface and to mediate the replication cycle of the virus. This is a more structurally resolved interaction of that ribosome binding, the receptor binding domain. And you can see individual amino acid residues, as they have been mutated here, are directly involved to mediate that higher affinity binding. So I used to not read not peer-reviewed preprint publications, but given how quickly data are evolving, I'm now looking at publications that have not even been published in a scientific journal, have not been peer-reviewed, but are in the public domain, so-called preprint publications. And there was one coming out a few days ago, and that published a mutation in the spike protein, pretty much in the middle of the protein here. And that mutation apparently is the reason why the virus succeeded to go beyond the province Hubei in China and become a global threat. The mutation seemed to have occurred very early on, so early in January, and is now becoming a dominant strain of the virus and might have enhanced its fitness. So apparently that mutation, it should not have been used or should have been considered in current vaccine formulations. If the mutation has not been considered, there might be concerns because it's also constituting an important epitope. So what the mutation apparently does, it's not really known, but it stabilizes the receptor binding domain up position. So its exposure to show enhanced interaction with the receptor. Also, it needs to be considered that there are epitopes at the surface. So epitopes are recognized by antibodies, so certain antibodies binding to those specific epitopes. That epitopes should not be considered in vaccine formulations because they are causing antibody-dependent enhancement, which is an immune enhancement which causes immunopathology due to an increased production of the virus. So it's contradicting the prevention concept. It's actually facilitating viral replication. So this is a major concern and needs to be seriously considered for all vaccine developments. There's a lot of detail in the next slides. So we are looking at what has been done with the SARS, the original SARS virus coming out in 2002, 2003. A lot of follow-up studies happened. And clearly the spike protein induced protective immunity. The RBD, that's the receptor binding domain of the spike, also induced immunity. The spike protein has ADE, these, detrimental epitopes. The RBD does not have those epitopes, so it would be advantageous. The RBD has been now favored as a vaccine subunit antigen for the development of a SARS vaccine. The spike protein and the nucleocapsid protein have been linked to enhanced immunopathology. That's the cytokine storm, that's the unspecific inflammation and destruction of tissue, which is causing enhanced morbidity and causes a severe development of the disease. We have been looking at patients who recovered from the disease. And it's clearly the cellular immunity, and in particular the CD8, the cytotoxic T cell responses, which are very important in patients recovering. They all have developed very good memory T cell responses. So these responses allow a quick recovery of the immune system if there's a new exposure and a better and enhanced and quicker defense. Neutralizing antibodies have been found against the spike protein, and also antibodies against the N protein and N protein were found. Strong T cell responses correlated with good neutralizing antibodies. So again, we have to be careful, not only looking for antibodies, but also recruiting cellular immunity, which was an important lesson. Now looking at COVID-19 patients and how is the response in those patients, patients recovering from the disease. And there's a nice, also coming from a preprint, medical archives, not published and peer-reviewed in a scientific journal, but you can see pre-epidemic individuals barely have any antibodies against the nucleocapsid protein or the spike protein, but if they are hospitalized, if they are diagnosed with COVID-19, antibody titers go up against the nucleocapsid protein and the spike protein. So clearly those proteins play a role in the immune response and in the defense against the pathogen. Unfortunately, antibodies are not long-lasting, and they're not cross-protecting anyway against the SARS. That's not too much of a problem if you're targeting COVID-19 itself. There is an immediate T cell response which is correlated with a mild disease, so cellular immunity. And if you have low T cell counts, mortality increases. So again, we need to balance between antibody and cell-mediated immune response, which is shown in this slide. So basically, we have the antibody response and we have the immune cell response. We need to make sure that we are producing plenty of antibodies which neutralize the virus, so inhibiting its infectious potential, its ability to attach, but also to label the virus for quick destruction by macrophages, for example, immune cells, which are scouting for labeled pathogens. So an ideal vaccine would mimic the immune response of the virus, but not causing the disease and cause a long-lasting protection in patients. The vaccine targets are clearly those proteins, and there has been lots of activities around vaccine development, and you see that in the news all the time. You can hear that, and I'm not going into detail. We have a lot of different strategies. A very promising strategy and one of the most advanced strategies at the moment is the RNA vaccine. And in the next slide, you can see that it has been just a few days ago approved by the FDA to go into phase 2. So it's messenger RNA formulated into lipid nanoparticles used for vaccination, and then the messenger RNA encodes for the spike protein, the S protein, but it's a stabilized S protein. Now these are all the clinical trials, and there are more coming through as we speak. There's a clinical trial pending that has been announced by the University of Queensland. It's also based on the S protein. This is a subunit vaccine initiative. So there's currently no existing vaccines, so coronaviruses are challenging. And we need, and that's a statement I want to make at this stage, so don't get too worried about all these different vaccine activities and vaccine approaches. I think the global pandemic, the SARS-CoV-2 caused pandemic, COVID-19, and its impact, its global impact justifies the development of multiple, or justifies the use of multiple vaccine development strategies. We have to go in parallel to mitigate the risk, as clearly many vaccine candidates will likely be unsuccessful. They will not induce protective immunity. They will have adverse reactions. They might induce antibody-dependent enhancement, immune enhancement, immunopathology. Immunity might be short-lived, which is then useless for a vaccine, and it might be costly. Scalable manufacturing might be prohibiting. They lack stability, which also adds to cost. So there is a strong argument for parallel activities in the vaccine development space. And this is showing you what CEPI is currently looking at and supporting. CEPI is the Coalition of Epidemic Preparedness Innovation. They have hundreds of millions of dollars available, and their mandate is to support platform technologies and rapid response technologies. So this is their definition of a rapid response. Within 16 weeks from antigen identification, a product should be released for clinical trials. 6 weeks from vaccination to clinical benefit, meaning immediate or quick establishment of an immune response after vaccination, and then 8 weeks to manufacture 100,000 doses. Our platform, within a week, we should have the synthetic DNA, and that can be shortened. In week 2, we would have a production strain. Vaccine candidates should be available in 4 weeks. In 8 weeks, we should have completed preclinical studies. And in week 12, we should be able to produce material for clinical trials. We believe, and that's based on our preclinical studies in the context of other diseases, that protective immunity can be induced within 6 weeks, and if not earlier. And productivity of our system, our platform, is unprecedented compared to other technologies. A 400-liter culture produces 4 million doses in 3 days. An estimated cost for production of a dose is less than $1. So now what we have done, and I'd like to emphasize again, Shushong Chen, Dr. Chen, he is working tirelessly 24/7 to make this happen. And he has taken DNA encoding vaccine antigens, and he has engineered our cell factory to produce those polymer particles coated with the antigen. And these polymer particles have been manufactured in the lab and isolated and purified. We are now looking at 4 different candidates, so different antigen combinations, different epitopes, just to cater for the desired immune responses. And we are now taking those into animal trials. But what would be the impact for Australia? There would be a rapid cost-effective mass production of the vaccine available if we are successful with our development activities, if the animal trials complete successfully, and that will enable quite quickly and timely extensive vaccination for immediate control of the outbreak. So we establish herd immunity. We will interrupt the possibility to spread the vaccine quite quickly. We will contain the outbreak, and hopefully life can go back to normality quite quickly. Queensland manufacturing capacity by itself would enable to produce enough doses to vaccinate the entire Australian population within 2 weeks. The first in human testing of our platform would be paving the way for the establishment of the platform as a rapid response technology for future preparedness. As I mentioned before, the platform is very flexible. If a new mutation comes through, if a new variant appears, a new threat establishes, we can quickly respond at scale. That's the current development status of our vaccine. We have completed a prime boost vaccination in mice. 70 mice have been recruited. No adverse reactions have been observed. Blood will be collected tomorrow, and we will have the immune response analyzed on May 20th. The next steps would require extensive funding. They're very expensive steps. I estimated $5 million would be required to take the vaccine material through toxicology to make sure it's safe, in addition to our animal trial data. And then the process needs to be transferred into a biopharmaceuticals production environment, a GMP manufacturing environment. We have a company here in Queensland, Luina Bio, with whom we have partnered to enable the technology transfer and the manufacture of clinical materials, which would then be tested initially in about 60 volunteers to confirm the safety of the vaccine. This would all be covered by an estimated funding of $5 million. So if you're interested to support the research and development of a COVID-19 vaccine as it is being conducted at Griffith University, please contact Olga, our development manager. If you have further questions, more technical questions, you are welcome to engage with me. My contact information is provided in this slide. You can also find me on Google. If you Google my name, you will see my affiliation and contact details. This is the website of the Center for Cell Factories and Biopolymers. And I'd like to thank you for taking your time to listen to that live stream presentation, and I'm happy to take questions. So just waiting for a few questions coming through the chat room. There was a question around animal efficacy testing. We're using mice as the first model to see immunogenicity of our vaccine candidates, to see what antibody responses, what cell-mediated immune responses we are inducing in the mouse model. We are also engaging with CSIRO in Australia in the Geelong facility to explore the possibility to go into animals which develop some kind of the COVID-19 disease. Those animals are ferrets. They can be vaccinated, and they then can be challenged with the virus, and establishment of symptoms or no establishment of symptoms can be correlated with protective immunity. So I'm just going through the questions. Yeah, there has been a question around what strains are currently more virulent and how territorial they are. As I said, one of the mutations, and people are now tracking mutations. There's a lot of omics research being done, genome sequencing. Apparently, the mutation, the D614G mutation, enabled the virus to go across the boundary of China and spread across the globe quite quickly. There's no real data and correlation about the severity and changes in severity yet caused by those mutations. There are mutations in sites where there's no obvious link to virulence, and there are also less frequent mutations. But clearly there's an indication that we are seeing mutations, that the virus is evolving, and so it's important to carefully monitor that evolution of the virus, also from a vaccine development perspective, but also from an antiviral therapy perspective, because it's a bit of a chase, it's a race between the virus and prevention and therapeutic strategies. That's why rapid response technologies are, in my opinion, very critical for this kind of threat, as we can quickly adopt changes. So if a new mutation arises and that's becoming more fit than existing strains, it will quickly spread, it will be detected, and it might constitute a new epitope. It might constitute an immune evasion mechanism, which then needs to be tackled by redeveloping the vaccine and adapting the vaccine, redesigning it. So there's a question coming through: how much vaccine can be produced by the cell factory? The cell factory has the major advantage that it can be grown in established facilities, and there are bioreactors available which can be as big as 100,000 liters. I mentioned 400-liter bioreactors, so you can easily look at 4,000-liter bioreactors, which would be 10 times of the 400-liter bioreactor, producing 10 times the amount of doses. So we are looking at 40 million doses produced in a 4,000-liter bioreactor, which is still, from industrial perspective, a small pilot plant bioreactor. So I believe, looking at global manufacturing capacity, there are contract manufacturing organizations. They have tremendous capacity available, that based on the cell factory concept, there should be the possibility of an immediate supply, global supply of the vaccine. The question is, why are there no vaccines against SARS or MERS? These outbreaks were quite quickly contained. They had a high mortality, they were contained quite quickly. Coronaviruses were pretty unknown before that time. The outbreaks in 2002, 3, and then 2012, the MERS outbreak triggered a lot of vaccine efforts, but there was no reoccurrence and no pandemic threat, so efforts slowed down. There is a strong activity around developing subunit vaccines in that space. Current vaccine efforts have not succeeded to produce a vaccine against SARS and MERS. Perhaps maybe there was too much focus on antibody responses. And the more we understand about the involvement of cell-mediated immune responses, the more vaccine development is considering this aspect of the immune response. But you also need to consider and keep in mind that the immune response is extremely complex. And it's an orchestrated mechanism involving subsets of immune cells and signaling molecules for a very balanced and fine-tuned response. And to orchestrate that so it's balanced and protects against the virus as opposed to causing damage by uncontrolled inflammation, that is very critical for vaccine design. And hence, again, it's important to use technologies which allow that tuning, that precision engineering of the vaccine to target very specific responses which are required to mediate a response and to mediate protective immunity. So it is and remains challenging. And as I said before, many technologies will likely not be successful, as it is a complicated immune response which needs to be established to protect against the virus. And we need to learn quickly from patients who recovered what immune responses mediated the recovery. And there's a lot of effort at the moment happening to understand what immunology is underpinning the protection against the virus. So I'm just looking at my see more. I'm just looking. There's a question. With so many other vaccines being developed all over the world for COVID-19, how likely is it that the vaccine being developed by you actually ends up getting used? That's a very good question. We are very hopeful because our vaccine materials are different from all the other vaccine materials. We have a unique composition of our materials. We are presenting antigens and formulate antigens in a very unique way. So they are taken up by those antigen presenting cells. And we know that they are effectively dissecting the antigens and display them at their surface to recruit the immune system. We have a good understanding what pathways are involved. That helps us to engineer the vaccine to favor certain pathways over other pathways, to mimic the response as best as possible to the response in patients who recovered. So having said that, by all means, I wish that as many as possible vaccine activities are successful. But by the nature of the complexity of the immune response required, it's unlikely that all of them would be successful. It's unlikely. The success is unlikely in general if you only look at certain aspects of the immune response. But the fact is that some vaccines are going into clinical trials, and that's a strong encouragement. The RNA vaccine going into phase 2 clinical trials is a very strong vote by the regulator in the United States that this could be a vaccine. So I'm hopeful that there is a chance that this could contribute and help to induce protective immunity. But as I mentioned before, there are major challenges for inducing protective immunity against this virus. Just there's another question: what makes your biopolymer platform better than other subunit vaccines delivery systems, and do you use adjuvants? That's a technical, that's a pretty technical question. So the polymer, and I mentioned that before, allows the presentation of antigens in a very particular way. The polymer also recruits particular antigen processing pathways in those antigen presenting cells, all in favor of very efficient loading of those receptors with viral peptides for efficient and strong immune responses. The material is different. It induces a different antigen processing pathway. It can be abundantly produced. It's very stable. And I also mentioned that before, we can quickly adopt and incorporate multiple antigens, which allows us to, for example, have the spike protein, the nucleocapsid protein, or the envelope protein, where we can find antibodies against in humans surviving from the disease, have them all together, maybe in different copy numbers to mimic the situation of the virus, and then induce the proper immune response. So having said that, the platform offers a huge flexibility for precision engineering other platforms wouldn't offer. So I got some messages here, so it's time to actually wrap up. Yeah, I really appreciate your time and your interest to listen to me. That's a live stream event, and we are very excited about applying our technology, and we're very hopeful that we can offer something unique to the vaccine development landscape. We urgently need funding to move to the next step. We're talking to the state, we're talking to the federal government, but yeah, so we are hopeful, and we're trying hard to provide a solution to this massive global problem. Thank you all for your attention and for the questions and engagement. And you will have my contact details, and I would be happy to respond and engage if you wish. All the best to everybody. Thank you.